So in this video, we're going to look at the higher level, the detail for DNA structure and DNA replication. The essential idea is much the same as we've met before. Structure, function, mantra. The structure of DNA is ideally suited to its function. The function, remember, is that you've got a code, and that code is one gene codes for one polypeptide. And the structure of DNA, as you'll see in 7.1 more clearly, is capable of being compacted into a very small space, especially in eukaryotic organisms. So these are the syllabus statements here. And again, can I draw your attention to the fact that I'm asking some of you to look at aspects of 7.2 in a little bit more detail and to present on the work of this. It's probably worthwhile, especially for this work on DNA, of getting a separate set of notes, which is pulling together all of the different experiments that have been done to support and to propose evidence to support the structure of DNA and how DNA is replicated. If we look at what Claire's doing, she's going to look at the work of Hershey and Chase. And Hershey and Chase were looking at whether or not it was DNA or protein that was responsible to be the hereditary material. We now know it's DNA, but back at this time they did not. This experiment is different from the experiment that was done by Messelson and Stahl. And in my experience, students very often get muddled up between Hershey and Chase and Messelson and Stahl. So signpost that to yourself in your notes and just make sure that you're clear on that work. This part here relates to what Ari and Angel are going to look at, and they're going to look at the technological advancements that are available to scientists now, and this idea of using molecular visualization software and looking at the differences again between protein and DNA. I'm going to introduce you in a moment to what this is meant by the nucleosome. Tanush and Surin, you're going to look at this idea of what we mean by DNA profiling. DNA profiling used to be called DNA fingerprinting, but this is used by forensic scientists. This is also a process which is used for looking at parent, looking at whether or not the child belongs to father A or father B. And what we're looking at when we're looking at DNA profile is something called short tandem repeats. So Tanush and Surin, I want you to start looking at what this means. Raphael and Catherine, you're going to look at this idea of di-deoxyribose nucleic acid being used to stop DNA and to try and elucidate the base sequence of a gene. This is a bit complicated. You might need some help with this one. And Kirsten and Alvin, you're going to look at Rosalind Franklin's work alongside Maurice Wilkins, and they looked at X-ray diffraction patterns, and using those X-ray diffraction patterns, we had a much more clear idea of how the DNA is arranged within that double helical structure. And this work complements work that we've already met about modeling that was done by Watson and Crick. So again, all of this idea of the nature of science, evidence is required to put forward and to maintain a particular model of the structure of DNA. This next slide, you can read it for yourself, but once we've looked at all of this work, it would be probably useful for you to go back and think about this. This is essentially talk stuff. So we're looking at how we know what we know about the structure of DNA, and we're looking at this idea of logical deductions from a variety of different experimental data to propose the model of the structure of DNA. So the nucleosome. In a eukaryotic cell, which you should already be aware of, are things called histone proteins. The Cognity platform actually has a very nice image of this, which I would suggest that you take a look at carefully. But remember that in prokaryotic organisms, the DNA was said to be naked. And in eukaryotic cells, it's not naked. And by not naked, it means that the DNA is wound around these balls of protein, which are called histone proteins. And if we look at this next diagram here, we can see the idea that the nucleosome, the whole structure here, is designed to protect the DNA. So the DNA is wrapped very tightly around this. And if it's wrapped very tightly around this, we're not going to mess about with the base sequence. We'll look later on at the differences between euchromatin and heterochromatin, but for now, I want you to appreciate that by wrapping it tightly around there, it adds stability to the molecule of DNA. We also need to recognize that histone is acidic in nature, whereas the DNA is basic, and that allows it to have this very, very tight connection here. So the DNA is packaged, it's supercoiled by being wrapped around here, and the linker proteins then will allow individual histones to align themselves back together. So if we look at this next slide, that's a little bit clearer. So we've got this linker DNA, and the linker DNA is allowing adjacent histones to pull together very, very, very tightly. What this diagram is also making reference to is that the nucleosome is made up of an octamer in the center of proteins. So we've got 8 proteins in the center here. Then we've got this histone H1, and histone H1 is also important in terms of the scaffolding and the stability of the molecule, and the linker DNA allowing one of these to connect with another one. And all of that allows us to package long lengths of DNA into a very, very short space. So we're going to package it. We've got this idea of it looks, once it's packaged, like beads on a string, and then we actually coil it up into what is shown here, which is the structure of the chromosome. So remember, in prokaryotic cells, there is no histone proteins. The DNA is naked, and you don't have the same degree of coiling up that is found here. Also notice on this chromosome, this is a chromosome with 2 sister chromatids, which means that this one has undergone DNA replication. So make connections here with what you've met previously. This next slide, again, is unpacking the same idea a little bit more, but the facts here speak for themselves. Okay, so you've got this idea that DNA in human cells is approximately 2 meters long. And if we look at the size of a chromosome, we can see that they are extremely small, and they are packaged together by this supercoiling. So the takeaways here is that it's eukaryotic DNA that's supercoiled. That allows the genetic material to be stored in a small place. It allows the genetic material to be tightly packed so you don't mess about and change the base sequence, because if you change the base sequence, then you can alter the expression of the genes. And that brings us on to this idea here. This will make much more sense when we look at the later work on transcription and translation. But if you want a gene to be expressed, you need this tight coiling to be slightly looser. So you've got a difference between heterochromatin and euchromatin. So what we've got here is this idea that euchromatin is tightly packed, and if it's tightly packed, then it's not allowing transcription, translation, and protein synthesis. In heterochromatin, what we've got are some aspects of the DNA molecule being slightly looser, and if there's a slightly looser connection here, that means that we can get RNA polymerase into this to do transcription, translation, and make the protein. But we only see heterochromatin if we've got specialized cells, because specialized cells have some genes switched on and some genes switched off. So again, signposting this back to the work that we've done previously. And the next slide is just underscoring the fact that when we do DNA replication, we're not starting at one end of the chromosome and going all the way along. We have multiple origins of replications, and that's important, and that's a distinction between the standard level and the higher level stuff. So we've got multiple replication forks being used at the same time, which can be seen here. So now we start to, this is the same thing again. So the origin of recognition, so the origin of why we start this replication here, and here is important. These are called origins of initiation, and they are happening because there are very, very similar base sequences for the enzymes helicase in the first instance to bind to. So what we've got is DNA helicase. We've met this before, but DNA helicase is binding at numerous sites, and it binds at numerous sites to unravel the DNA and separate the hydrogen bonds that exist between the DNA. Difference here between standard level and higher level is the fact that we've got multiple origins of initiation taking place, which we didn't look at for standard level. In the next slide, what we see again is this idea of the new strand being produced by polymerase 3. It's working in this direction. But what we need to do here is consider the differences between higher level and standard level by looking at 2 different forms of polymerase. We've got DNA polymerase 3 and DNA polymerase 1. Now, it's DNA polymerase 3 that is responsible for doing this addition of the nucleotides into the right position and making the phosphodiester bonds here. But again, for higher level, we need to think a little bit more in depth, and these are not just nucleotides. I alluded to this in the first video. What these are something called nucleoside triphosphates. And I'm going to jump a couple of slides down here to show you what I mean here. So DNA polymerase 3 is adding new nucleotides in the 3' to 5' on the 3' end of the parental strands, so that the new strand is made in the 5' to 3' direction. But when we talk about DNA polymerase 3, instead of talking about a new nucleotide, we want to talk about a nucleoside triphosphate. That's an error there. That should be nucleoside. Let's correct that right now. Pardon me. And if we look at this diagram in more detail, we can see that what we've got is the sugar bit, we've got the base bit, and we've got the phosphate bit here. So if we cut these two off, we would have a nucleotide. So a nucleotide is when we've got a sugar bit, a base bit, and one phosphate. The nucleoside refers to the sugar bit and the base bit. So the sugar bit and the base bit is referred to as a nucleoside. So we can have a nucleoside monophosphate, which is equal to a nucleotide. We can have a nucleoside diphosphate, and we can have a nucleoside triphosphate. And in this case, we've got what is referred to as a nucleoside triphosphate that is being brought into position. And the advantage of doing this is that when we bring this into position, and remember that the base is going to be aligned with the parental base, which is going to be complementary. So we've got cytosine, guanine, adenine, and thymine. When this is brought into position, what we can then do is cut the bond here. We splice that bond, and in so doing, we release a lot of free energy, and that's the free energy that allows us to construct the phosphodiester bond with the adjacent nucleotide. So we've got a difference in the triple material here, which is saying that we need to appreciate that what this enzyme is DNA polymerase 3, and DNA polymerase 3 is bringing a nucleoside triphosphate into position. And we can have deoxyribose nucleic acid, and remember we can have ribose nucleic acid, which is what this is about there. But I would use the term nucleoside triphosphate. So if we look at, again, add in a little bit more detail again, in order for DNA polymerase 3 to bind, it needs to have a mechanism to attach itself to the right place. So what we need to appreciate here is an extra thing that is omitted from standard level, and to appreciate that we have a short section of actually RNA primers are there so-called. So RNA primase makes an RNA primer, and it pops it onto the end of the molecule here. And remember, this is going to be the 3' end of the parental strand, so that when we make the new strand, it will work in the 5' to 3' direction. So what we need to add here is the idea that DNA helicase is going to unzip the molecule. You will read also about another molecule called DNA gyrase, which I'll come back to in the summary. But DNA helicase unzips it. Then what we've got is RNA primase attaching an RNA primer to one of these exposed parental strands. That primer allows for DNA polymerase to bind, and then DNA polymerase is going to add nucleoside triphosphates using complementary base pairing, and it's going to work in the 5' to 3' direction towards the replication fork on the lead-in strand, because it's going all the way from the 5' to the 3' direction, and on the what's referred to as the lag-in strand, we've got discontinuous replication by DNA polymerase 3. But again, in order for DNA polymerase 3 to join on this lag-in strand, we need to have, first of all, the insertion of a primer. The primer then allows for DNA polymerase 3 to bind, and then DNA polymerase 3 is going to make this section. Okay, then there's going to be another primer, and DNA polymerase is going to work its way along. Now, these fragments that are made are referred to as